Welcome everyone to our YouTube channel. Today's theme of discussion is a database, that is GeneCards. Today's discussion will include a brief description of GeneCards, its features, application it holds, starting with GeneCards, input and output results, and some research project ideas. So, starting with a brief description about GeneCards is a comprehensive integrative database that provides detailed information about human genes. Launched in 1997 and developed by Weizmann Institute of Science, GeneCard aggregates data from over 150 distinct sources, including genomic, proteomic, transcriptomic, and functional information. The database aims to facilitate quick access to a wealth of information about genes, their function, associated disease, and relevant biological pathways. Each gene entry is presented in a user-friendly card format, making it easy for researchers to find and interpret the information they need. To understand better about GeneCards, we should go through key features and properties of GeneCards database. First, comprehensive gene information. It covers over 73,000 human genes, providing extensive annotations and data. Secondly, it is integrated data source. It merges information from more than 150 databases including NCBI, Ensemble, and UniProt. Then, its user-friendly interface. It offers a simple and intuitive search interface for easy navigation and information retrieval. Then, gene-centric format. Each gene is presented in a card format that consolidates various types of information, including functions, pathways, and diseases. Then, batch query capabilities. It features tools like GENECARD for batch querying multiple genes simultaneously. Then, disease and pathways integrations. Links genes to associated diseases and biological pathways, aiding in biomedical research. Then, regular updates. The database is updated periodically to include the latest research findings and data enhancements. Then, tools for analysis includes tools like Gene Analytics for gene set enrichment analysis and GeneDex for finding similar genes. Then, access to external resources provides direct links to related databases for further exploration of gene information. And community engagement. It is used widely by researchers, clinicians, and educators, with millions of users accessing the database annually. Moving forward to the diverse application of GeneCards database in research, we have gene function annotation, understanding the roles and function of specific genes in various biological processes. Then it offers diverse disease association studies. It helps in investigating the genetic basis of diseases by linking genes to the associated disorders. Then, pathway analysis, exploring metabolic and signaling pathways involving specific genes. Then, drug target identification, identifying potential drug targets by analyzing gene-disease relationship. Then, comparatively genomics, comparing gene information across different species to study evolutionary relationships. Then, transcriptomic studies, analyzing gene expression data to understand regulatory mechanisms. Then, proteomic research, studying protein interaction and functions related to specific genes. Then, SNP analysis, investigating single nucleotide polymorphism and their association with disease. Then, biomarker discovery, identifying biomarkers for diseases through gene expression and functional studies. Lastly, educational resource, serving as a teaching tool for students and educators in genetics and genomics. Now, let's understand how a beginner can start his journey with GeneCards. First, access the database. Visit the GeneCards website at www.genecards.organization. Then, familiarize with the interface. Explore the home page and familiarize yourself with the search bar and navigation options. Then, conduct a simple search. Enter gene name or symbol in the search bar to retrieve its card. Then, explore GeneCards. Click on the gene entry to view detailed information including gene function, associated disease, and pathways. Lastly, utilize tools. Experiment with tools like GENECARD for batch queries and gene analytics for enrichment analysis. Now, let's go through input and output results description. First, input. Users can input gene names, symbols, or identifiers, examples UniProt SSN number, into the search bar. Advanced search options allows filtering by specific criteria. Then, output. The output includes a detailed gene card that presents information such as gene summary, functions, pathways, related diseases, and links to external databases. Users can also download data in various formats. Here we have presented research project ideas using GENECARD's database. First project idea is investigating gene diseases associations. Its steps include Step 1. Select a disease interest, example breast cancer. Step 2. Use GENECARDs to identify genes associated with breast cancer. Step 3. Analyze the GENECARDs for functions and pathways related to the disease. Step 4. Compile findings into report discussing the implications for treatment or further research. Second project idea is pathway analysis of a specific gene. Its step includes Step 1. Choose a gene, example TP53, known for its role in cancer. Step 2. Search for the gene in GENECARDs and assess its pathway information. Step 4. Map out the pathways involving TP53 and identify interactions with other genes. Step 4. Present your analysis in a visual format such as a pathway diagram. Third and last project idea is on batch query for comparative gene analysis. Its steps include Step 1. Identify a list of genes related to a specific biological process. Example, apoptosis. Step 2. Use GENECARD to input the list and select relevant data fields. Step 3. Retrieve and analyze the output data for functional similarities and differences. Step 4. Summarize your findings and propose hypotheses for the further investigation. I hope you all have found this session insightful and have got a better understanding of GENECARD's database. Join our training session and project to enhance your understanding of the GENECARD's database and its application in research. Like and subscribe for more educational content and feel free to comment if you have any queries and suggestions for future video topics. Thank you all.